We have seen aerobic and anaerobic respiration as means to produce ATP. We're now going to look at the third method for making ATP, fermentation. Fermentation is used by organisms that cannot undergo respiration, whether because they don't have a transport chain or because there is no oxygen to undergo aerobic respiration. In this case, ATP will be produced only through glycolysis. The location of fermentation in both eukaryotes and prokaryotes is the cytoplasm. The purpose of fermentation is to use pyruvate or a derivative of pyruvate as the terminal electron acceptor for NADH. Fermentation produces NAD+ for glycolysis. That is, fermentation oxidizes NADH back to NAD+ so that NAD+ can be reduced at glycolysis. There are many different types of fermentation pathways. Fermentation pathways are named after the end products of the pathway. In general, we can state that bacterial fermentation yields acids. In general, we can state that yeast fermentation yields alcohols. Acids and alcohols are end products of fermentation. End products of fermentation include compounds like lactic acid, ethanol, butyric acid, propionic acid, 2,3-butanediol, mixed acids. Remember, fermentations are named after their end products. Lactic acid fermentation yields lactic acid. Ethanol fermentation yields ethanol as waste products, etc. Here is one of the simplest forms of fermentation, lactic acid fermentation. Remember, by definition, fermentation is a process that uses pyruvate or a derivative of pyruvate to oxidize NADH back to NAD+. So remember that during glycolysis, a glucose molecule is oxidized and transformed into two pyruvate molecules. In the process, two ATP molecules are produced and two NADH molecules are produced through the oxidation of glucose intermediates. If the organism has an electron transport chain, the NAD electron carriers will be oxidized by the electron transport chain as the electrons are given to the carriers, producing NAD+, which can go back to glycolysis to be reduced once more. But what happens if there is no electron transport chain? If the only mechanism to make ATP is glycolysis, NADH accumulates until there is no more NAD+ available to oxidize glucose. The purpose of fermentation is to oxidize NADH back to the NAD+ form so the NAD+ carrier can go back to glycolysis and oxidize glucose derivatives once more. In lactic acid fermentation, pyruvate receives the electron of NADH, oxidizing the molecule and converting it back to NAD+. Pyruvate is converted to lactate when it picks up the electrons from NADH. Ethanol fermentation is another form of fermentation. In this case, the molecule that oxidizes NADH is not pyruvate, but rather a derivative of pyruvate, acetaldehyde. Acetaldehyde will oxidize NADH and it will be converted to ethanol. So remember, fermentation is a process through which pyruvate or a derivative of pyruvate, in this case acetaldehyde, oxidizes NADH to convert it back to NAD+. Thus, NAD+ is again available to oxidize glucose and its derivatives so that ATP can continue to be produced through glycolysis. There are many different types of fermentation. In general, bacterial fermentation produces acids. For example, E. coli fermentation produces a mixture of acids, acetic acid, lactic acid, succinic acid, and some alcohol. On the other hand, another bacteria called Enterobacter produces a mixture of acids plus an alcohol called butanediol. We call this type of fermentation butanediol fermentation. Another organism called Streptococcus and another genus called Lactobacillus undergo lactic acid fermentation. Propionibacterium undergoes propionic acid and acetic acid fermentation. Clostridium produces butyric acid, butyl alcohol, acetone, and isopropyl alcohol as the products of its fermentation. Saccharomyces is a yeast. It produces alcohols as products of fermentation. Notice that the products of fermentation could be used to identify organisms. For example, both E. coli and Enterobacter are gram-negative rods belonging to the same family, the Enterobacteriaceae family. They are both lactose and glucose fermenters. The difference between them is that E. coli fermentation produces a mixture of acids, whereas Enterobacter fermentation produces a mixture of acids plus alcohols. For example, the methyl red Voges-Pasteur test, or MRVP test, can distinguish between the mixed acid fermenters like E. coli versus the butanediol fermenters like Enterobacter. In this biochemical test, the organism that is being tested will be incubated in a medium that has glucose. The organism is allowed to ferment for 24 or 48 hours. After fermentation, methyl red is added to a portion of the medium to test for the presence of acids. A positive MR test indicates the organism is a mixed acid fermenter like E. coli. The other portion of the medium is tested with a reagent that will detect the presence of butanediol. If butanediol is present, that will indicate the organism is a butanediol fermenter like Enterobacter. Thus, organisms can be identified by testing for the presence of the end products of their fermentation. As a summary of what we have been looking at in both Chapter 4 and Chapter 6, remember strict aerobic organisms make energy ATP by aerobic respiration. Strict anaerobes make energy by either anaerobic respiration or, in some cases, by fermentation. Facultative anaerobes can make energy by aerobic respiration in the presence of oxygen, or fermentation or anaerobic respiration in the absence of oxygen. On the other hand, aerotolerants only have fermentation as their means of making ATP. Out of all these mechanisms, aerobic respiration is the more efficient. followed by anaerobic respiration, and last, fermentation. The only mechanism through which fermenters make ATP is glycolysis. Therefore, they will produce only 2 ATP molecules per glucose. You should now be able to fill the metabolic pathways chapter 6 chart with the information regarding fermentation. You should know the location of the pathway in both eukaryotes and prokaryotes, the starting reactants, the final products, the number of oxidation steps, if any, the number of ATP synthesized, the number of decarboxylation steps, if any. A microbiology student is trying to identify two organisms, A and B. Both are gram-negative rods, facultative anaerobes. The student performs the MRVP test, methyl red Volks-Pasteur test, on the organisms with the following results. MR test is positive on organism A and VP is negative. MR test is negative on organism B and VP test is positive. Which of the organisms is E. coli and which is Enterobacter? Put the following pathways in order from most efficient to least efficient: fermentation, aerobic respiration, anaerobic respiration.